Okay. We're going to talk about the adaptive immune system here. So remember how I talked about the linking of the innate to the adaptive immune system and how a cell called an antigen presenting cell will present that antigen to another cell. That's where we left off. Remember that? So over here we have a macrophage that has taken in a microbe, like a bacteria cell, processed the antigen, it's projecting it on its surface. Actually, it's projecting it on a MHC2 molecule that's like the little flag that it wears to let other cells know that it's a self cell. And it kind of sits there and it finds a cell to present it to. Notice how this is a helper T cell and it's inactive. Helper T cell, another name for is a CD4 cell because that little blue candy cane looking thing is the CD4 protein on the surface. This other cell surface protein it has is a T cell receptor. Okay? So the first step is essentially there is a connection there when the antigen presenting cell presents that antigen to the helper T cell, and the helper T cell then becomes active. Okay? So when the T cell is activated, it can do something. Well, actually, many different things, but it starts this process called clonal selection. It starts creating clones of itself, essentially, and they differentiate. Some of them become memory CD4 cells, so they remember that antigen that's encountered. Memory cells are more long-lasting. Okay. It also can become active CD4 cells. These are the effector cells. And I added on there this little arrow here. That just means that once active, that CD4 helper T cell secretes something called interleukin-2. Your book talks about more details of other co-stimulants. I just wanted to focus on interleukin-2. So it just starts releasing it as it's moving around your body. It's going to help down here in the other responses to make a connection stronger. Next, let's talk about the two branches of the adaptive immune system. It can either be cell-mediated or antibody-mediated. Cell-mediated are, think of the cells are doing the work. The cells can leave the lymphatic system, go find cells in the body that are infected and essentially destroy them. It's cell-to-cell combat, essentially, is what cell-mediated anatomy, or not, adaptive immune system is. Antibody-mediated, just like the name would imply, produces antibodies. Okay, so let's talk about cell-mediated here. Here we have a body cell, an infected body cell. So it's been infected with this microbe that was over there, okay? And the antigen of that is right there. So what cells in your body do is they also project that antigen on their surface, on their MHC, in this case, MHC1 molecule, to tell other cells in your body, hey, we need help, essentially. They are waving their antigen, saying, help, 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 okay? And then the cells that look for them are the cytotoxic CD8 cells. Notice that it's inactive to begin with. Cytotoxic cells also have a T cell receptor, but they're called CD8 cells because they have a different protein on the surface. So it recognizes that antigen, and after it recognizes it, it becomes activated. But to increase that activation stimulation to essentially make it stronger, this is when these helper T cells come in and they help. They help make that essentially connection stronger and pretty much just saying, yes, we need to react to this antigen. We need to mount a strong immune response. So the co-stimulation here is what it's called, as this CD4 cell releases interleukin-2. It's just a chemical that makes that signal stronger. Once that happens, that cytotoxic T cell is now active, and it is going to make more of itself. That's called clonal selection. And it can either make active CD8 cells. These are the effector cells. So we can write effector. Any cell that's going to go do the work are called effector cells. In this case, CD8 cells, cytotoxic, they go find cells, other cells that are infected, and they kill them. How they do it, they release a chemical called perforin that will essentially punch holes in the microbes or the infected cells, and then granulozyme will come in and help just essentially make it explode, destroy it. They can also make memory CD8 cells that, of course, remember that same antigen up here. Okay, so that is cell-mediated. Over to antibody-mediated. Notice we have B cells over here instead of T cells. This one is an active. It also has a receptor. This receptor is called a B cell receptor. How does it get activated? It's a little unique that to become activated, it can actually recognize an antigen on a microbe itself, okay? And it can be activated that way through its receptor. Again, just like on the other side, the T cell receptor recognizing the antigen right here on the MHC molecule can make that a stronger message saying, yes, we need to mount an attack by the release of the interleukin-2. Okay? So it's just making it a stronger message, more of a stronger response. So now the B cell is active, and it can start to make more B cells. It can either make memory B cells. These are more